Okay, there we go. It's recording.
So we have this program called Team Viewer. It's a remote desktop app. So we are using this to log into the ddPCR computer. Do you use TeamViewer usually?
Yeah, the ddPCR computer.
The TeamViewer software. Have you used this before?
I have used it, yeah.
Okay, nice. Seems like someone else has done a ddPCR. I don't know who it is, so let me double check. Wait, so you did a few columns of qPCR, right? ddPCR?
Yeah.
Okay, this is somebody else then.
For the last run?
Yeah.
Yeah, we had 3 columns, but 1 column was just the no template control.
Okay.
Yeah.
So it should be this one, I think, the 39. It has 2 samples and 1 column of water.
Yeah, that looks like it's, yeah.
So,
Mm-hmm,
so this name, the control_water, I've been putting this for the samples that have the vaccine spiked into water and then concentrated. So if you're doing a no template control, I would name it like this. I would just name it NTC underscore any dummy thing.
Okay.
Does it make sense?
Yeah. Mm-hmm.
That's what I've been following so far.
Okay.
So let's maybe close this file. I don't know who has done this. The last 2 qPCRs have not been logged. They have been done today. Okay. So here we go and say low. And it makes the screen up. You notice how there are 2 things of the same name for the 39.
Mm-hmm.
So one of them is an empty file and the other one has a lot of things in it. You can see the size here, it's 13,000 kb. The other one is 146 kb.
Yeah.
So when you start the machine, sometimes it gives you an error, and then you have to abort the run, and then it makes a dummy file with this folder with the same name, but not much files in it except the error file.
Okay.
So whenever I see that, I go and delete it just to avoid confusion.
Yeah. Okay.
But make sure you're deleting the correct file.
Yeah.
So I just want to double check this data, see if it has worked. So you go and select analyze.
Mm-hmm.
Seems like there are a few droplets here and there.
Okay. And it seems like most of them have nothing.
Yeah. What samples are these again?
I don't know what samples they are. I think they came. Was it Thursday?
Okay, so these are the manhole samples then.
Yeah. Yeah.
So they collect some extra samples and bring them to us sometime in the week.
Okay.
So these are the wells with no template, right? You see a few droplets coming up here and there as well.
Yeah.
Yeah, so I don't know what's the explanation for that, but Lauren always gets concerned whenever I show her the data.
Yeah. When it like some positives show up in the blanks.
Mm-hmm.
Yeah.
So I select the data that I am interested in. We'll ignore the water things for now. And click on the status. It will show you all the ones that have not been done properly. So we'll manually go to each of these and we can double check the thresholds if they are set properly. Alternately, it seems like right now there's a clear separation between the positive and negative, right? You can set the threshold for all the samples at the same time. If there is no ambiguity, you can just click here and then it creates thresholds in both the directions and then changes all the status to manual.
Okay.
So we'll just go with that.
So how do you know where to place the threshold? Do you just kind of like
yeah, so currently we are just eyeballing it, but the goal is to have a big cluster in this third quadrant and the fourth quadrant and the first, second, and fourth. These three quadrants should have clusters of points.
Okay.
And so you don't want to set a threshold that cuts through any of these clusters, especially the large black one. So you'll see the number of points here are so much higher than the other ones. If you go to the 1D amplitude, it shows a histogram here on the right. So all of these points here, those are the black ones.
Okay.
So the small number of points at 7,000, 6,000 the amplitude, they don't have any density here. So there's very few points that are positive.
So I have another question. For when you set the threshold, why, let's say, for example, if you set it at the 3,000, like, because you have 2,000, 3,000, what if you put it at 3,000, 3,000? Like, I guess what difference would it make?
Okay, so yeah, let's see this point and try to figure out where it belongs to.
Oh, okay. Oh, okay, I see now. That's why you...
So only for that one particular sample, it will make a difference.
Oh, I see.
Okay, so it seems like it is G1. So we'll just scroll over to G1. Okay, we can just test it on G1 for now. So G1 is mostly negative, it seems like. So here you observe the values 0.17 and 0.09, and then I'll change the threshold, and then we'll observe the values again. So change the value for N1. we made it 0.
Oh, okay.
So usually when a point is this far from the negative, I think it's usually considered to be positive because there's
Yeah,
no other explanation why the intensity would be so high.
I see. Okay.
Unless it's a stray
Makes
point
sense.
very close to the negative clusters. So like these two here, these are ambiguous.
Yeah,
We wouldn't know how to classify them.
I see. Okay.
So for example, if in a certain sample you see points here, but you also see some points here, then you would consider these as positive and these will definitely be negative.
Okay.
But if a well has only points here, then you could think of putting the threshold closer. But
Okay.
I don't know if there's a hard and fast rule for that. We're
I
still trying
see.
to figure out how to work that. But you can see one point here.
Mm-hmm.
That means that's a normal droplet fluorescence.
Mm-hmm.
If that well has huge load of inhibitors or something, then it might
Mm-hmm.
bring this whole point down to here. But that
Mm-hmm.
doesn't generally happen.
Okay.
So make sure you select all of the points and then
Okay.
you do an export CSV and we'll overwrite this file. So the original file will have the automatic values, but the few of them, they didn't have any values because they had errors due to the automatic processing.
Mm-hmm.
So previously the points were a little more ambiguous, so the separation between clusters. So that's why I
Right.
handpicked the few ones that weren't done automatically, and only those I used to manually threshold.
Okay.
So make sure you check all of the wells are in here. So if you selected only a few, if you selected only this column and clicked on export, then it would export only that column.
Okay.
So we have this information. We will copy this whole thing
Mm-hmm.
and we will paste it in this thing called raw ddPCR. So I'll create a new sheet for each new file and I will paste it here and then I will name it according to whatever is in the sample notebook. So I'll just copy the name from here and then I'll put it as the sheet name.
Okay.
Alright, so this data is already in, but there's also another folder where I've been storing these CSV files just for like in case I want to double check if I made an error or something. I have the raw data handy.
Mm.
Like it doesn't really do anything in the analysis, but I've just been doing it.
Mm-hmm.
So that part is done. Now you want to make sure that you save this actually. So all the manual thresholds that we did, they will not be saved unless you click on save. Or unless you exit the software, it will ask you about saving. So if somebody has been playing around with the threshold, trying to see if their samples are working or not, but they don't really want to save it, you want to exit the software and click no in that case. If you leave it open and someone else comes onto the team viewer, they won't know what to do with that open file.
Okay.
So that's why I've been wondering what those 40, 41 samples are and who they belong to. And there is one that is mislabeled as well. Looks like a real sample to me. You can check. So you can check the size. If the size is in certain megabytes, then it's a real folder. Otherwise, it's an empty folder with an error message in it. If it's in kilobytes. So we are done with the droplet PCR stage right now, and we can close TeamViewer. So then we can go to the qPCR part. You can go to any Google Drive folder, type qPCR. and it will take you to this folder. And I've been saving all of the files into this COV to wastewater work.
Mm-hmm.
If you don't have access to this, then you can click on that request access, and then it will send it to that computer that's running the qPCR. So whoever goes on to that next, they will accept it.
All right.
So from here, we can download the latest files that I have run. So I will download this one. This is the same concentration control. Oh, so this is the one that Basma ran. Okay, people haven't been adding all of their runs onto the sheet, it looks like. Okay, so we are interested in this 713. So we'll go here and I'll download this onto my computer. And so this file format is a proprietary thing by Thermo Fisher or Applied Biosystems. Okay, so my files ends at 70, 71, 72. So this is the next one in the series. So I'll open this in this proprietary software that they give for free. If you don't have this on your computer, you can also do this in TeamViewer. So in TeamViewer, so if you want to log into any of these and you don't know the TeamViewer login information, you'll go to the super user sheet. So if you don't know how to get to the sheet, again, you go to any folder in your Google Drive, type super user, and it will show you this sheet, Stadler group members and other stuff. So inside the sheet, I've put in the computers as well. So let's say, where's the qPCR computer? Here it is. So you scroll to the right, you will see TeamViewer ID and password. This is for the qPCR laptop. And same for the ddPCR, you will find a different ID and a password. Alright, so this file has opened up. You will see that there is nothing has amplified, which is a little suspicious. Also, the ddPCR looks kind of lower than usual. So we'll get the, once we process the file, we can look at the graphs and see if anything looks completely off. We'll have to redo these 12 samples again, 16.
So nothing, so the ddPCR file gives you like, I saw up there it said copies per microliter.
Mm-hmm.
So it gives you that, but you don't yet know if it's been amplified until you put it into the software.
No, it is amplified. So the droplets that we saw, that is what is generating that copies per microliter. But it seems lower than usual.
Okay.
So I'm just wondering if there was some mistake during setting up the run or something. Usually in ddPCR, if you see droplets and if you see that it has run properly, there should not be any other issue. But the qPCR has not worked at all. So definitely.
Oh, this is this is the qPCR. Okay.
Yeah. So the qPCR, you don't see any amplification here.
Right. Okay.
So I generally check the raw data as well to make sure that the data analysis has. Okay, so the standards, even the standards didn't amplify. So definitely there's something wrong with either she didn't add the probes or something.
Yeah.
And there's one more quality control step in the ddPCR. you can look at this in the software as well.
Okay.
I forgot to show you, but in the file that you export, there is many columns. There's a lot of columns. So the main thing is what we're taking home from here is this thing, copies per 20 microliter well.
Mm-hmm.
And so the value that we saw, the copies per microliter, that is calculating for a 20 microliter reaction volume. But we only added 10 microliter of template into that. So we do that manually to calculate the copies per microliter. So I take this value and then I divide it by approximately 10, which is the amount of template we're adding. It's not exactly 10 because we made excess of the reaction. We made a 22 microliter reaction. So into that 10 microliter of reaction went of template. So this will be 9 point something.
Yeah.
So I set up that inside my R script. So it does that automatically.
Okay.
So inside this, this is the thing step where there's another quality control. You want to make sure the number of droplets, accepted droplets, is greater than 10,000. So if it's too low, the software generally flags it, and that's when you'll see the status as check, because it will ask you to check if the droplets actually look good or if they are spurious. And some of them have very low droplets, 1,000, 2,000. Those samples I generally discard them, because when you do the statistics on here, they have this confidence interval, max and min. Sometimes the confidence interval is so large because the droplets are so low that you want to discard the data rather than threshold it manually. Does that kind of make sense?
Yeah. So you would discard the data that has the high confidence intervals, the white.
Yeah.
Okay.
I generally don't look at that, but I just look at the number of accepted droplets.
Yeah. Okay.
So we'll go back to the qPCR. Let's assume there's some real data here.
Mm.
And so what you would do is to go and export. You will change this file name to, so this is the thing. I set up my R code in a certain manner. So I have a folder, I'm calling it qPCR. But if you clone this GitHub files, you will get a few of these. Yeah, this is a different branch, so there's way too many scripts. Okay, this is the latest branch, and you'll see fewer scripts in this, I think. Yeah, so the main thing is 1, 2, and this and that. So these are the 4 main scripts. There's another one that's also involved. So you want to keep all of these, and then there's this R project file. If you open this, it will open directly from this folder. So you don't need to change your working directory inside R. If you open through this, it will directly open all of these files as well. So within this, there is a certain organization here. The Excel files that I export from this machine, they should be saved onto this directory. And so this is all inside my box.com cloud. So you don't need all of these old files. So if you're just analyzing something new, just make any folder you want and to download the Git code there, but make sure the subfolders are named the same. So you want to make a folder with, what's this?
And do you have this shared on your box?
Which one?
Because
The code.
own box folder, right?
Yeah, I think my box folder is not shared with everyone. I shared it only with David, I think.
Oh, okay.
So there's a lot of files in here. If you need access, then I can share it with you as well.
Okay.
But so the thing is, Zach made it work even without this whole thing. All you need is to download this this file and make sure that you're exporting the data into a subfolder that's called Excel files. So this could be any folder that your script is in, and the subfolder is called Excel files. And make sure to make another subfolder that's called qPCR analysis, because it's the code is set to make graphs in this. If it doesn't see any folder, then it throws an error.
Okay.
And the raw files have been storing here, but the code doesn't see this, so it's fine.
Okay.
And the name, so this is the trick. I put this information in this in the README as well. If you open up GitHub, the first page that you see, you'll see a README here. So here I wrote a step-by-step guide of all the things that you would need to do. I think this is not the current branch. I haven't merged it into the main thing yet, but it's called formula_show.
Okay.
So here is the naming convention. So you want to name it with this, the batch ID of the qPCR file, and then the whatever description you want to give your samples, and then the target name, and this is the ID of the standard curve, and it goes in a serial order. So if you're not sure what order you are in, there's this file where the data is being saved. It's called data dump. So inside this, you'll find a sheet called standard curves. So the last used standard curve will have its ID here. So it's STD53. And the one in my folder is also the same, standard 53. So what I do is I copy this name from here and then add the standard to it. So it has everything except the standard. So I'll add 54 to this. Does that make sense?
Yeah, that makes sense.
Yeah, so I will take that and then just say export, and then it makes a file here. I don't know if I want to export this data. It looks real messy. There's nothing amplified, so there's no use exporting this. But I have another file that has been exported. So let's, I'll show you how to analyze this 915 data, the one from last week. So last week's data should be here, this one. So you want to copy this name, and so in R I made up this pipeline where you make a vector, let's say file names vector. That's too long. So you make a string with this ddPCR thing as one of the names, and the other name will be the qPCR file of the same thing, which is this thing, 71. And it has to include the standard and this as well. If you miss any of the, if you miss the target name or this, it will throw up an error. So you put both of these into a vector. You can also process them individually, but this is just the workflow that I have. So let's go to, so open Quant Studio and then export the Excel file. Everything else is written there. We already did the ddPCR thing. Check to make sure that we will do this one step ahead. We will do this later. So we are going to RStudio and we made a vector with the two names that we want to process. And then we run this command that says map, and it's calling this function to run on each element of this vector. So the actual function that this is running are there in this script, process functions.r. So you want to source this file first before you run the command. So what I like to do is I just keep this checked and I save the file and it sources, but there's also a source button there. So it takes a little while when you run it, but then this is what we do is map, and then what did we name our thing as? You can go to environment flnms. So this is the vector and this is the name of the function. So I have independent processing functions, one for the standard curve, one for the rest of the qPCR data minus the standard curve, and the third one is the ddPCR data. And so I just set up a common function where it looks at the file name that you're giving it. If it sees a dd.ww, then it calls this ddPCR function. If it sees a standard in the file name, then it calls that. And if it doesn't see any of those, it calls the qPCR function. So once it runs the standard, it will also go to this next step and run the qPCR. But for using the standard curve data, it's calling, so it's taking the data from the sheet. So that's why I've been saving the data. As soon as it runs, it saves the data here. So it calculates the slope and the intercept and efficiency and all that. Also saves the data on a folder here. Inside qPCR analysis, yeah, you need to make sure there's another folder called standards. So it is going to save the image there. And this is how it looks like. So you want to double check to see this image and make sure your standards are proper. So for example, if we took this, okay, let's actually export this data and see what it looks like. So I will paste this name here, and then we'll also paste the ddPCR thing. It's here, right? The 39. So I paste these two things, and then I made this different vector, and then I'm going to call process all qPCR, and then it will ask you for your Google account that you want to access, and then you just, if you have already entered it, you just select the old thing. So David has made a different way of using this. So instead of using your own account, he made a common account for the COVID wastewater project. And so the advantage of using that is that there's no limits on the quota. So if you simultaneously back to back make calls for 10 different sheets, and then Google throws an error thinking that you are a robot trying to steal its resources. So if you make your own Google Cloud account, then you don't have that problem. Okay, there's an error in fitting because there's no data in that file. Alright, I should have thought of that. So this is going to work. I will just delete this and show you with the old data. Okay, so for now, what we can do is just run the first one. We'll just run the ddPCR for now. So if you are running just one thing and you know it's already a ddPCR, you can directly call this process ddPCR, and then you give this name. And before you do that, but we'll do it while it's running now, you want to go here, make sure these names are proper. So 916 1A 1B. So what are these 1A 1B?
1A, 1B. I'm not sure, honestly. I don't know much about the manhole samples. I just helped to extract them and then helped with the quantification.
So you got 4 bottles, right?
What was that?
You got 4 bottles of this sample.
Right.
So I'm assuming you did 2 replicates of each bottle.
Yeah, I believe so. I didn't concentrate the samples.
Okay, so the way I have been naming, let's go and stop the, no, this has already run, that's fine. So the way I've been naming my samples.
All of those are separate, yeah.
So I want the date to be, or the sample, the batch number to be in the middle, and the actual sample name to be on the right. So for this, I would rename this as 916 AO, right? So this is how I want to name my samples. If you name it in any other way, then it doesn't make the graph properly. So you want to make sure to check this before you run this first script. I'll make sure to put it into the README. So there should be a guide here on how to name your samples. Maybe I didn't update it yet. We'll check that later. Okay. So and you'll notice once I corrected this, even this is corrected because I've been using a formula here instead of pasting the actual things. I write a formula replacing the N1 and 2 with B curve. So if you make a change in one place, it will reflect in other things too. So we'll just rerun this file. When you run it for the second time, it will ask you about overwriting the old data. And if you're not sure what you're doing, then you should say no. But right now we just change the file names. So we want to say yes. Once that has run, it will make a new sheet here with this name. So you'll observe how it has partitioned the name. The 916 or NTC came here, the AO came here, and there's biological replicate 1, 2, 3. That's the thing after the dot. And so the first step of the processing, it takes both the qPCR, which is here for the old sample for 915, and the ddPCR data. It will take both of them here. And then what you'll do is you'll go to the second script. So I should have this written down, I think. Okay, so you'll open the second file and you'll change a couple of variables in the beginning, and then you can run that script. So what that will do is we can actually rerun this old data, or we can, so what it's doing is, if it's all of these are the same samples, it's taking the ddPCR data for that sample and the qPCR for the same sample, it stitches them together, and then it plots them overlapping with each other so you can compare easily. And so if your samples are distributed between multiple runs, this is a good way to run the sample. So I just make a vector, and then I say bring all of these things, and I use the same map command again. So it's going through this vector and bringing all the sheets, and then it jumbles them together. It uses this bind rows to mix them together. So if you're aware of the R, and have you used deep layer inside R?
Yeah, I have.
Yeah, so that is the framework I've been using.
Okay.
The tidyverse deep layer framework.
Yep, I use that. Yep.
Yeah, so just make sure you give it the names here, and you change this title name. So for our thing right now, we only have this file.
Mm-hmm.
So I'll delete all of the old ones and I'll put this. And you want to change this because otherwise it will overwrite the old data. So this is 916. And so this is the sheet where the IDs that we write on the portal and the actual name of the treatment plant, they are linked in this sheet. So what we do is, the photo that we take in the treatment plant, previously the health department people, they used to just make it into an Excel sheet, and I just copy paste it into this Google sheet here. But nowadays they are busy, so I do it manually myself. So each week has an entry here, and then there is this column name, which is the things that we write on the portal. This is the short ID, and this is the full name of the treatment plant. So the code is going through this, and it's grabbing this data, and whenever it sees this as the name, so the name that we are giving it, it has, so it has 916 SOH. No, this is not a good example. Okay, so we have ABCD on the bottle, so it has 914B1, let's say. So it goes through this sheet, looks for 914B, and then it attaches these two things to that same entry. And so for that, you want to make sure to update this every week for different samples. So for the manhole samples, we don't have them in the sheet anyway. The sheet is only for the wastewater samples. So for the extra samples we get, the name already has the identifier in them. So they don't have ABC in their names. They have this full name, or an abbreviated form of the name. So it will just make these as the names, and it will ignore whatever is here. And we will, okay. So if you're running any control experiments, then you want to make this false. Otherwise, it will output the data into two other extra sheets where you don't need the data. So right now we need this data to give to the health department. So I put this as true. And once you change those three things, you are good to go and click on run this file, and it takes a couple of minutes to run. But you'll see what it's doing. It's going to grab each of these files that you mentioned. So this file that's in the data dump, it is grabbing that. And the second one that's from the biobot IDs, it is going to grab that. And there's another thing in sample registry called concentrated samples. I'll show you what that is. So it's reading from data dump. There's more things that it's reading. There's a vaccine summary. So if our samples were spiked, then we have the spike ID written on those bottles. The exact same spike ID is stored in the registry here as well. So I'll go to one of the recent samples. So the recent one was 914915 and the stock ID is S33. So when it processes the qPCR data, if it recognizes something that's named vaccine, so that's why what you write in this is very important in this table. So here you'll find something that says vaccine. So if there's a spelling mistake in this, or if it's written in the wrong column, if it moves around, then the program is going to fail. So it takes that vaccine data, and it's also saving it in another temporary file here. In this data dump, there's a thing for vaccines. So it takes the data, there's like multiplicates of the same thing. So there's S18.123, and the other sheet, which is vaccine summary, it averages that 123. So for S18, there will be only one entry here, like that. So the recent one is S33. So the data for that, it will be grabbed from here. So it has grabbed all of those things, and then it writes a couple of outputs. So one standard output is this other sheet called complete data. So the difference between this and the data dump is this thing has additional information incorporated in it. I forgot to mention it. So for each sample name that's being written in the registry, we have the stock ID of the spike. We also have the weight of the bottle they measure. And then they estimate it to be density of water, and they give us a volume. So the volume that we pour into each bottle is variable, but the amount of virus we are spiking is 50 microliters. It's the same. So if you want the concentration of virus, we will divide by this volume. And then, so there's the amount of volume they extracted of wastewater. So 50 ml of wastewater goes into the concentration thing. And other information is just for record keeping. But the code is looking at these four things. These three things. And it's also looking at this column called biobot ID. So previously I just used to use this column and remove the one or something like that. But then sometimes we have a few extra samples. They have a E or something like that included in the name. So this is the same as 915J1, which is already here. So you can't repeat the same name twice. So what people do is, this is an extra sample that's been processed for a different experiment. Usually they write those in the comment. Yeah, inhibition test. So this is for a different purpose, but it's the same sample. So you included an E here. Okay, this thing should not have an E. I should tell people. So these kind of things are where the code fails. If people don't write the registry properly, then the code fails because of that. I haven't processed any of these data, so we are good for now. But let's make a note. Priyanka. When it has gone through all of this data, it collected them all together. Then it does the calculation. So it divides by the volume and it does some other things. So it takes this virus, the vaccine ID that we give it, and then it takes the amount of quantification that we have for that, and it accounts for that. It does some normalization. A lot of things is happening here. So the information will be in this. So when I write my code, I try to divide it into segments, and that way you can access it easily through that button there. So it's doing multiple things. So it's dividing the virus concentration by the volume of virus that is spiked in, which is always 50 microliters. So it is hard coded here. Spike virus volume is hard coded here. If it changes for any particular occasion, like for a smaller tube, if you added a smaller volume, you'll have to either manually just go to that Excel sheet and change that calculation yourself manually. That's the easiest thing to do, because it's using this value for all the samples. So if you change for one, it will change for all of them. And it's calculating a recovery, which is the amount of virus, the bovine virus that we detect, divided by the amount that we have spiked in theoretically. So it's doing a bunch of other calculations as well. And it takes all of the data and then it makes plots. So there's multiple data that you can plot from here. So instead of saving graphs individually, what I do is I call another script. It's an R markdown script. So it takes in values like this. I can show you the script here. So the last thing that this script does is calling this function, that's something.rmt. And this function, it opens up the data and then it does plotting. And I've written some functions to make the plotting more standard as well. All of these additional functions are in this general functions.r. So there's this thing called plot, mean, standard deviation, jitter. You'll see how the plot looks like when you see. So this is the one that we ran just now. So the name that we gave here, the title name for this file, that is the name of all the file names that it outputs. So 916 manhole. That's the same name here. That will be the same in the sheet that it gets saved into. I'll just open this one. See if the data looks interesting. Okay, that is very interesting because these samples were not added to the registry. It could not find the volumes for these. So do you remember, did you do the concentration for these samples or was it Pawan?
It was Basma that did the concentration.
You mean extraction?
No, I think she did the concentration too.
I thought Pawan was doing it on the same day.
Oh, you know what? He did do it. Yeah, you're right. He did do the concentration. Yeah.
Okay, let's send a reminder.
Yeah, that's true. I think Basma did the concentration. I think there were some samples that came in over the weekend, and I know she said she did it for that. I was getting confused. So basically, all of the results, the output from the code goes into this file.
All the what? Can you repeat?
The output from the code that you just ran, it all goes into this report.
Yeah.
Okay.
It all goes into this report, but these are only the graphs and the data will be stored here.
Okay.
So the raw data, which is copies per microliter RNA, is here, but there's a fold concentration between going from wastewater to RNA. So that is what this volume accounts for. So what we can do is to compare from a previous week. Let's say 907. So we are looking at Star of Hope and Acton Oaks, right? AO and SOH. No, these things are not here. There's the jail, and okay, let's just do this instead. So there's another output which is wastewater treatment data for health department. So the difference is this file, it includes, so you'll notice now the file is named with the name of the treatment plant and not the name of the sample like this one was. Like the ones here where you don't find this name anymore. Yeah, so ABCD, you won't find these names anymore. You'll find the name of the treatment plant. And if a certain place doesn't have the name of the treatment plant, it will just name it whatever was there. So for NTC, it just names it as the NTC. And there's some samples that are water. So for the second sheet, it's filtering out all of those things that don't need to be given to the health department. And this sheet also partitions by, so there's the actual samples, which is the wastewater, and there's special samples, which is just the jail and the other, all the manhole samples that we receive. So these are the 915 and these are 916 that we just ran. Okay, so we'll see the copies per microliter RNA is 0.2, 0.3. You can check previous weeks. Okay, we have styrofoam here. So 3, 5, 6, 8. So 2 weeks earlier it was around 10-fold higher. Now it is 10-fold lower. So I don't know if it's a real decrease or if it's a qPCR didn't work. Okay, so for AO it's around 12, 11, 10. But now it is, oh, it's 0.2. So that's a huge decrease. And for styrofoam, there's a lot of things that are non-detect as well. I think it might be worthwhile to redo the ddPCR on this. So this time I can do it and you guys can watch again to see where we might have gone wrong.
Yeah, or
most probably this data is correct and we'll see the same results, but just to double check, because it's only 2 columns, we can redo it again.
yeah, or maybe we can do it and you can just watch to make sure.
Yeah, sure.
Yeah,
that
yeah,
would be good too.
yeah.
And also, I don't know what these 1A, 1B are from your initial naming. So it seems like you have 2 technical replicates for each bottle. So for example, here, the G1 and G2 that you see,
Yeah.
those are 250 ml samples from the same bottle that we take, but they are processed in 2 different concentration runs and they stick to 2 different filters.
Yeah.
So in the case of these samples, where are these? So in the case of these, we already have 4 of those, 1, 2, 3, 4. But then there's this A and B. So I don't know what those are. Are they like the same sample extracted on 2 different RNA filters? Not RNA filters, on 2 different columns with the RNA kit or something like that. So it's a replicate within a replicate.
So so from the extraction standpoint, we didn't make duplicates of what was already there. It was just one was labeled A, one was labeled B, and et cetera. So there was no like duplicating any of those.
Okay, so when you got the samples, they already had A and B on them. So maybe it was Pawan who did these things twice.
Maybe, yeah.
That's why if people put these things in the registry immediately, it's easy to trace back what your results are coming from.
Yeah.
Also, if you send this data to the health department, they will ask you why there are like so many replicates of the same sample.
Yeah.
Okay, so this is where the data is being saved. And if there's any other wastewater samples, you will find another sheet that has just 916 manhole, let's say, without the special sample.
Okay.
So if I put this as the name 915 rice, the actual samples are here, but special samples are the manhole samples. So it just adds that string to this thing and makes a different sheet.
Mm.
So when the wastewater treatment, so the health department, when they look at the data, they want them in two different sheets. So what I've been doing is in Excel files, I maintain this weekly data. So the last week's data that I gave them, it has two sheets, and they're pretty particular about naming the column names. But for us, in our case, the column names are being given programmatically, so they will be consistent all the time. So they are using another program in R to read these files and then compile these from Rice and Baylor. So if you mess with anything, then the program will crash. So we have the wastewater things here, and other location has the manhole stuff, the jail and the ODM, all these things. So what I do is I just copy the data from here and then I paste it into what am I saying, Excel file, and then I save it as .xlsx. So I don't use Office products, but I use a different software, but it gives the same interface and it works the same way. And there's another thing which I haven't run for this week, I think. So we can run it right now. So once you get this data, so this is the data for one particular week. But if you wanted to compare the data across weeks, you would run another script called weeklycomparisons.r. So here I have a long running string. It has the names of all of the things that you want to include in this data file. I was going to do one thing before I ran this code. So the last time I ran this is how the graphs look like. So you'll see the recovery fraction is it's the percentage recovery. I tried to name it that way, but the percentage sign, it throws the things off. So I just named it this way. Maybe I should just write in words, percent recovery or something, so it's more clear. So for one particular week, it was, this is some error happening in the data processing. That's why it's going up to 1000%. So that's the reason why you can't see any trends in other weeks, because this graph is so zoomed out. So I'm going to remove this 805 and then rerun this data. So I will remove it.
So that's in units of percent.
That's in percentage, yeah.
Okay.
So I will add the 915 rice to this. And so right now, this program, this is one of the things that will throw off the Google Sheets pipeline, because it's going to load 10 different sheets and back to back. So Google thinks that you're trying to spam its resources. So if you're lucky, it works on the first run. Otherwise, we have to use the method that David created, which I haven't figured out how to use yet. So we'll rename that title, just make it 622 to 915 rice. We can write no 805. It was it 805. Okay, that's fine. And then there's other files that this is loading. So there's this metadata file, and this is, what is this bringing? Okay, so this is bringing the data of the population in each treatment plant. They have this file where they save that, And they also have the flow rates for each week. So what they're doing is they're kind of normalizing this. So certain treatment plants are large, certain other ones are small. So there's two very big treatment plants, and all the other ones are very small. So they have a very different flow rate. So if you normalize to that, you can account for the size of the treatment plant. And then if you divide by the population, you will have a per capita number of viruses, something like that. So this code just brings those data, and then it will attach it to this data with all the weeks in it. Let's run it and see if it throws an error or anything. Okay, I'll walk you through this file just in case. So the first thing is the dot plot of the recovery. Second plot is the exact same, except it has a violin plot laid on top of it. You can't see anything right now because this is very zoomed out. Third plot is another way to show the recovery. So first three plots are showing recovery. The subsequent ones are showing you the data copies per liter wastewater. And so this thing has all three in them, the N1, N2, and B curve. You can see that B curve is almost always higher than the N1 and 2. So if you want to see data in close, I remove that, and then this has only N1 and N2. You can see that there's a few outliers. Most of the samples are here, few outliers are here. We will have to investigate later why certain things are higher than the others. But this is the same plot. You can't see the things that are low. So if you blow it up in a log scale, you can see more detailed information. And there's many more plots giving you similar kind of information. So this is N1 versus N2 to see if there was any significant bias within that particular week. If there's something wrong with your qPCR setup or whatever, this is one place where you can catch it. But as of now, all of them seem to have the same kind of trend line. Also, when you change the method or something, you'll generally have a change in this correlation between N1 and 2 as well. And now these are very tiny plots. What they're doing is at each treatment plant, it's connecting the dots for each time point. So x-axis is the time points. I used to have fewer number of weeks previously, but Lauren wants all of the data here. So this is what I've been doing. But I think there's no trend that's visible. So for this time, what we can do is next time we run it, we can remove all of these old ones. Just run it August, mid-August through September or something like that. And the other file that it was outputting previously, the other HTML file. So I broke up the data in multiple segments. So this is most of the data. Again, there's a lot of things, so the x-axis is very crowded. You can't see much. I don't generally like labels being inclined, so I avoid doing that. But when I have multiple categories of data, so this, for example, the 914 and the NTC, these are the information that you entered here. You'll see the between the dash and the underscore, that's where the 916 is coming from. And in this case, the NTC is coming from there. So if you named it in a different way, like you previously had AO and N16, then it wouldn't be able to plot this thing properly. So right now, all the 914 are categorized under one thing. So that's what I'm aiming for. With if the data labeling is streamlined, then your code will do all the processing. And so this is the per liter wastewater. If there's any missing volumes here, you'll see the data for them here, but you'll not see data here. That's when you know you'll go back to the registry and mess with them, message the people who know that information and ask them to enter it as soon as possible. So then this is the recovery. Okay, so this is just the B curve. It's the same plot as above without the N1 and N2. And there's the same thing but percentage recovery. So here I have open circles showing you the calculated amount of virus that we spiked in. It's dividing by the volume. So that gives you the number of molecules of BCO that was there. But the one that you, after extracting, when you quantify, this is the actual quantification. So that's the expected and the actual. If you divide those two, then you have a percentage recovery. So right now we are seeing around 20%. This is the same thing but on a log scale. And then if there's additional samples, usually the first plot gets too crowded. So I thought I removed the NTC as well, but I usually put NTC and other stuff into this plot here. So that way you can see the side experiments or anything else that you do, controls, vaccine and stuff, you can see them clearly here. There's more scatter plots that are plotting N1 versus N2, but all the time they have some errors, so they don't work. Interesting. So previously the code used to stop there abruptly because there's an error, but now I made like a error catching function where if you see that there's some error, it just outputs this thing and say it skips the rest of the thing in that function. So that's the advantage there. And these are the same plots as above, but arranged in an ascending order of the red points. So you can see that if there's a clear trend, if there's a certain treatment plans every week having a high N1, N2 or whatever, you can compare this graph across weeks to see if these things remain the same or are they switching around. Or we'll see the same thing with this once this is done running. Weekly. Alright, so you can see more of a trend here because I removed that one outlying data. You can see a certain weeks the recovery fraction is increasing and then it has some cyclic trend or something for some reason. So there's a few weeks that are outliers.
I see.
And the reason for this is the vaccine quantification that we are using. I think I had a thing of that in the lab notebook.
So why is it above 100%?
Why is it above 100%? This
Like
is
the
a
recovery?
question that I get a lot. So there's two things that we are missing. The vaccine that we're quantifying using this extraction method, it seems to be inefficient in itself. So there's this vaccine boiling. So I think I walked through this data before in the lab meeting. But when they do an extraction using an RNA extraction kit, using Kiagen, they were doing it, they see a much lower value of RNA molecules compared to when we do the boiling and lysis that I showed you the other day.
Mm-hmm.
There's a huge variability between the lysate method, probably because it's so crude or something, or I don't know why that would be the case. But you see that it's consistently higher. So we are using the blue values for calculating this. So if it happens that it's the red values that is true and not the blue, so all of your data will be adjusted downwards because you are going to be dividing with a higher number. I assume that will take those values below 100%. There's another inaccuracy in the pipeline. So I'm using a concentration fold from wastewater to RNA as 1,000-fold because I'm putting in 50 ml wastewater and I'm taking out 50 microliter of the RNA extract. But there is a few steps in the middle where they were throwing out a little bit of the sample. So if I account for that, it's actually 500-fold, not 1,000-fold. If I adjust for that, even that will take this a bit down.
Okay, I see.
So those are the two explanations we have as of now.
I see.
So one thing that's clear is the last 2 weeks, there's a significant going up trend in the recovery. I wonder why that's the case. Right, do you see that, the red points?
Yeah, I can see it.
Okay, so the recovery fraction seems to be consistent, but then the actual data points is going up.
Yeah.
This is the not unnormalized thing.
Like the concentrations are going up.
Yeah. I wonder what's happening. So we just like look through each plot, and these are exploratory data plots.
Mm-hmm.
So there's also another function where you've used ggplot before, right?
Oh yeah. Mm-hmm.
Have you done any interactive plots so far?
Interactive plots? No, I don't believe so. Not that I can think.
Okay, so I'll show you one. There's this package called Plotly,
Mm-hmm.
Plotly, and that has a very nice function.
I've heard of
It
that.
takes any ggplot that you make. So, for example, let's see if this works. No, this is not the function that I'm going for. Where's the weekly comparisons? Okay. I guess even this can be fine. So I have another function that says format log scale y. The thing that it does, it converts the y-axis to log scale, but it also makes the axis labels in a particular way. So I've done a lot of background work to put all these together. So the simple thing is just scale y log 10. I think this many people do. But then I found another function to do this formatting of the y-axis. And I just called it a fancy scientific, that's the name of the function. And it has a lot of elements to it. So if it's a 1 into 10 power something, it removes the 1 because it's redundant. But if it's something else, that it's a 3 into 10 power something, previously it used to output as a 10 power 0 point something something, 10 power 0.3. That's not useful. So I made that thing go into the bottom of the exponent. So all that processing is taken care of here. So I'll call, instead of printing this ggplot, I will call plotly and then ggplotly. This is the function. I'm writing this in the wrong place. I should be writing it here. And so instead of the plots, it opens this in the viewer now. And the cool thing about this is an interactive plot. So you can uncheck a few points that you don't want or something like that. You can zoom in. Yeah, so there are a bunch of stuff that you can do. If you want to analyze a certain data more closely, you can do things like that. Or if you want to analyze only the blue points, you will turn off all the red points. Something like that. So you can do this when you have a high dimension. For example, for this one, so it turns out that these things that have the plus command on it, you can't change them. So you've used this symbol before, right? For piping commands in R.
Yeah, I've used that before. Oh yeah.
Yeah. So this and plus, they don't work well together. So you have to
Yeah.
put things in flowery brackets.
Mm-hmm. Yeah.
So let's say you had this uh, you had this data and you wanted to focus on 3 particular weeks. So you could do that. And if you double click, it goes back to home state. So I guess the problem is this thing is not completely set up to do the same thing that ggplot does. So the function here, it was distributing the droplets left and right. So that is being done by the jitter function. But this thing doesn't recognize that. So it gives them all in a straight line. So it's not as useful. So yeah, that is the basic overview of the data analysis. Do you have any other questions? So I haven't gone through the checklist, but
Yeah.
I put all of these individual things in here.
Yeah. No, I think it's pretty clear. And then I have the recording to go back to refer back to. And so what you send to the health department are like the output Excel files from the script?
Mm-hmm.
Okay. Okay.
Yeah, so there are multiple outputs from the script. One is for our own purpose, which has all the controls and any other additional experiments that we do.
Yeah.
The other one has only the wastewater data.
Okay. Okay. Yeah, I think that's pretty much. I'll probably maybe look at some samples that were already done just to kind of go through the whole thing.
Yeah, so the thing is, when you're rerunning any old data, if you want to see the output, it will ask you about overwriting the existing files.
Yeah. I'll just, can I rename it so that it's not going to do that?
Yeah, you can rename your files as a testing or something like that and then put the other name. So that way it will not ask you for overlapping names.
Right. Okay.
But if you run the first step of the process, the file name that it's grabbing from and the file it's output into is the same name. So you might want to rename your Excel file as well.
Okay.
To have the testing or dummy or something like that.
Right, right. Okay. I'll do that. All right. Well, thank you. Thanks for showing me that.
Yeah, you're welcome. And also the recording that I have walking